Okay, good. Thank you very much, everyone. Dr. Reinhart.
Thanks. Sorry about that. So thanks for being here. Thanks for the invitation. Like Roy said, I'm run a lab. We're interested in the neural basis, neuromechanisms of various components of visual perception and cognition. And I was trained as a basic scientist at Vanderbilt with Jeff Woodman, Jeff Shaw, Sohee Park, but maybe 1/3 of the lab projects also involve clinical populations. And so I'll have some teaser data about that throughout the talk, but mainly it's going to be healthy young people, healthy older people. These days, our research streams that are supported look like this. We have a lot of other stuff happening in the lab. And so we're interested in kind of neurocomputational mechanisms of memory in a healthy case, as well as how those systems break down in memory disorders like Alzheimer's and mild cognitive impairment. We'll also have research happening in the realm of executive functions, like working memory updating and inhibitory control components like this, as well as how they break down in people with schizophrenia. And then we have another stream on reward processing, decision making, broadly construed, and also people with OCD. Today, given Roy's background in neurology, I thought this would be most appropriate to focus on some of our latest memory research. And so, you know, in our lab, we one of the unifying themes is trying to get some insight into neurocomputational mechanism through brain oscillations and how they synchronize. So, you know, whether we're trying to formulate a theoretically meaningful question or design a new neuromodulation protocol, we get a lot of our inspiration. The work is often grounded in the fundamental neurosciences, system neuroscience, which views cognition as arising from synchronous electrophysiological oscillations or rhythms in the brain across different spatial temporal scales. So, right, oscillations are these repetitive patterns of brain activity. They derive from inhibitory and excitatory postsynaptic fluctuations that are evident in the cyclic changes in the LFP, ECOG, EEG, MEG. More relevant is their purported functions. So just from bird's eye view, it's often fairly well agreed upon that oscillatory synchronization, these fast neuronal correlations, may serve a role for brain communication and plasticity. And so they're thought to facilitate the sculpting and modulation of neurophysiological dynamic spikes, kind of coining the realm in neuroscience, right, for how information is represented in the brain. And so it's thought that with oscillatory synchronization, there could be a facilitation of precise but flexible brain information exchange and communication that could promote flexible adaptive behavior. So if you have synchronization, it's the idea is that you have like a gate is open or like excitability window is open, and so you get more increased likelihood that information can flow within and between brain regions, as opposed to when there's low synchronization or a gate is closed. In addition to coordinating spike timing, the synchronization of brain rhythms is also shown to induce the spike field coherence, spike time dependent plasticity. It's really for these reasons that it's thought to really represent a more like fundamental cognitive neural process. What's fun now is that we potentially have these new tools in our utility belt as researchers that can non-invasively augment, act on these putative signatures of mental function, these oscillatory synchronization signatures, that leverage the importance of timing in the brain, these time-based mechanisms. They leverage the importance of the kind of rhythmic qualities of brain activity and allow us to potentially kind of unprecedented control over modulating rhythmic activity patterns in target regions implicated in cognitive function and dysfunction. The tool transcranial alternating current stimulation that we have at our disposal that could potentially manipulate these rhythmic patterns of brain activity. It's a technique in which sinusoidal alternating currents are applied to the surface of the scalp via electrodes, placed non-invasively, and induces an alternating electric field gradients in a target region. So they're, allows for the possibility of potentially modulating these macroscopic brain networks at the single units level. You can apply TACS to the surface of the scalp in non-human primates, like here with an electrode advanced down into the brain, and you can see how it changes spike phase or spike timing independently of spike rate. So this is I'm sure you're familiar with it because you have Desmond Oates and Roy Hamilton, all these amazing brain stimulation researchers, but this is a sub-threshold neuromodulation approach. So unlike TMS, we're not inducing de novo firing. This is extremely weak levels of electrical current. And you can see, as you'd expect, there isn't a change in the amount of spikes per unit time. In animal models, what you see instead is the modulation of spike phase, the timing, so you can more temporally organize the spikes in the TACS period. Unlike in the sham, where there's just a lot of heterogeneity over time in their spiking profile. And you can even see this parametric manipulation, which came out later, where you increase the intensity of the stimulation, you can see a corresponding increase in the temporal organization of the spikes with TACS. This is one of the kind of leading action mechanisms of TACS, neuronal entrainment. Another one similar to this sub-threshold neuromodulation approaches in general is these neuroplastic mechanisms. There's beautiful animal literature going back at least to the 50s, where really this pioneering work was done by like Bindman and colleagues with direct current stimulation, suggesting that LTP and LTD are the underlying mechanisms for those long-lasting after effects. You can inhibit protein synthesis, for example, and see those after effects go away. I'm thinking of, like, for example, if you stimulate the forepaw of an animal and you're stimulating whether it's primary visual, primary somatosensory, primary auditory cortex, you see these beautiful increases in the number of spikes over 10-second bins, increases in evoked activity, spontaneous activity, and quiescence and decrease of activity when you flip the terminals of this battery around and apply, in this case, cathodal stimulation. There's a lot of lovely literature again, supporting this view that mechanisms of TDCS lie within at the cellular level, LTP, LTD here. Just showing an example with TACS in humans, whether you can use an NNBA receptor antagonist and abolish a TACS after effect, in this case, motor beta power, suggesting that this kind of the effects are mediated by synaptic plasticity, NMDA receptor mediated synaptic plasticity. There's other research to this effect. It's just kind of coherent with neuroplasticity as being another action mechanism for the long-lasting after effects of these subthreshold approaches like TDCS, TACS. There's a number of new advances in TACS. I just want to highlight real quick. They're going to be in the studies that I subsequently show you. One is this higher resolution approach. So you could see in the old days, the more conventional approach, these larger sponge pads, maybe 5 by 7 centimeters, and they kind of take out a large swath of cortex here in the current flow model shown to the right. but now we have much smaller electrodes. We have an array of electrodes, and we can arrange them in the sync source center surround configuration where it can increase the focality, anatomical targeting of the stimulation. So obviously you can get these 4x1 designs, for example, with an anode flanked by 4 cathodes, kind of funnel the current into your target region, which is nice. This has opened up more opportunities for targeting multiple regions simultaneously, which sometimes we call synchrony modulation. We're trying to target a long-range stream of phase-based connectivity. The lovely thing about this technique, again, it really capitalizes on timing and phasic relationships between neural populations that could be segregated, spatially segregated cortical regions. And we can do that because we have taken this high definition approach with smaller electrodes and many more of them. So here we can stimulate not just a single region, but multiple regions simultaneously and inform the stimulation design from this kind of neural network neuroscience perspective on which particular cortical regions are involved in how are they communicating with each other, because we can manipulate phase information between these regions. And then another advance that researchers are using is personalizing the stimulation to each individual in a lot of different cognitive, neural objects of inquiry. There's a significant amount of inter-individual variability. And we've been personalizing along frequency lines, individually tailoring the stimulation to the peak frequency of someone's functional electrophysiology or connectivity measure. Other people are personalizing along anatomical lines. But this is definitely one of the key features of our research. We're finding some evidence that it might be an essential feature to see the kind of effects that we're seeing. So as a kind of petri dish test case for some of these ideas, a few years ago, we were thinking seriously about cognitive aging and working memory. I've been doing working memory through grad school and now as faculty. So kind of combining our core expertise in working memory and executive control with this newer domain of aging that we were getting excited about applying some of these ideas. And I like to show you what we are finding. So we know from cross-sectional and longitudinal studies that there are a number of abilities that remain pretty stable and preserved across the adult lifespan, which is nice. Abilities like these 5. But we also know there are various capacities that show this rather sharp linear decline with increasing age. One of those is working memory. And that's going to be the focus here. Here's just some examples of classic working memory measurements across decades of life. Again, you see this kind of robust linear decline in healthy people. It looks a little different in pathological aging. It kind of has this S shape to it. And so working memory is something that we think is, really one of these fundamental cognitive building blocks of information processing architecture, right? It allows us to hold behaviorally useful information in mind over a brief time scale, and it's used for a wide variety of different processes and real-world applications like planning and decision-making, performing mathematical calculations, language comprehension. It's a place where we hold information, but also obviously to kind of work on that information. So it's often conceptualized, right, by cognitive psychologists as a workbench or a sketch pad of the mind, a seat of consciousness. It's just really one of these essential abilities. So we think it's very interesting. And for a long time, and we still don't really know how information is represented in the brain, right? It's still one of really basic question. We don't know how information is actually represented and carried forward in time. But a lot of the work has focused on this memory process within the working memory, the maintenance process within the working memory system. There's other processes that are not shown here, but these are kind of the canonical ones. And the question is like, how do we hold information in mind when it's in the absence of sensory input? And just to take you all the way back, it's an idea that's still fairly prominent these days. Maybe it's wrong, but it's the Hebbian cell assembly, right, from the late 40s. And Donald Hebb proposed this model for how neural firing can be sustained over these brief timescales. The famous finding that you find in the animal research from like Goldman Rakesh and others, where I'll show you in a moment what that looks like. But this toy model here is just showing the Hebbian cell assembly, where you have a group of neurons that fire, and then that triggers the firing of another group of neurons, and on down through the circuit and then back up again. So you get this sequence of reactivating cell assemblies, creating this like closed, this dynamic loop of activity. And maybe this, it was proposed, is how you are able to carry information forward in time when it's no longer viewable. So here's just some classic examples of what you see in systems neuroscience. Again, kind of unifying theme here is for our brain stimulation, for our theorizing, and in the human cognitive neuroscience, we often look to systems neuroscience for inspiration, for understanding. And here's just an illustration of this. So here's just a classic delayed match, classic oculomotor delayed response task. So it comes in like this. You fixate a central point. You're a monkey. Often there's a cue in the periphery. You learn to hold that peripheral target in mind over a delay period when it's no longer viewable. And then you're cued to make a saccadic eye movement to the remembered location. You get a juice reward or water reward. I did some of these experiments with macaque monkeys in grad school with Jeff Shull. And what you commonly observe is here, this is actually human work, I believe in parietal cortex. And here, but it really comes from the monkey neurophysiology. Here what you see commonly is spike field coherence in the gamma band. So you're seeing spikes that correlate with a particular phase interval in a systematic fashion of the LFP in the gamma band, roughly 50 hertz. You see something, so here again, the LFP is potentially like helping to organize these spikes in a regular way. Here's another example, but from lower frequencies in hippocampus. You can see the groups of neurons are organized, in this case, on the trough of the theta rhythm, this lower frequency oscillation. These are just example observations of how rhythmic activity fields, whether it's low frequency like theta and alpha or high frequency like beta and gamma, facilitate the formation of coherently organized groups of neurons via the establishment of transient temporal correlations, via the establishment of neural synchronization. This is not new stuff. It's just a little background if you're unfamiliar with it. If we pop up to the level of cognitive neuroscience with sulci and gyri and networks, a larger spatial scale of analysis that we do in human neuroscience, there's connections. It's a lovely thing about working with neural oscillations. The interface is nice with animal neurophysiology and lower spatial scales of analysis like fields and spikes. And so here, if we pop up, we see an interesting similar relationships here between low and high frequency. There's a particular neurocoding scheme, neurocoding motif that theta gamma phase amplitude coupling, it's a mouthful. Theta gamma PAC, phase amplitude coupling, is something you reliably see here, pre-surgical epileptic patients with an electrode advanced into temporal cortex. You see a parametric manipulation. As you load more objects into your working memory, you see a parametric increase in theta gamma phase amplitude coupling. What is it? It's when the amplitude of a fast wave like gamma down here in this cartoon is correlated to some phase interval of a slow cycle, a slower rhythm like theta up here. In this case, it's peak to peak relations. It doesn't have to be that kind of organization. So it's been proposed in the working memory literature that theta gamma phase amplitude coupling may be a neural signature for ongoing working memory process and storage of the individual feature, storage of the objects in these sensory processing regions. Another region that's commonly implicated in working memory literature is the prefrontal cortex, and there more current theories propose that it's not storage per se, but the prefrontal cortex acting as almost like a fulcrum that reaches back to those sensory regions and controls, monitors, coordinates activity. And for that, there's another coding motif. These are probably the best well-studied neural coding motifs, or however you want to call them, in systems neuroscience: theta gamma phase amplitude coupling and theta phase synchronization. Theta phase synchronization is often, although there's new evidence from the Pulva group that suggests otherwise, but overwhelmingly it's commonly found that lower frequencies may carry information across longer distances in the brain, higher frequencies maybe for more local processing. Theta phase synchronization is where you have these rhythmic activities, kind of related, these cyclic oscillations that are correlated in time, and usually within the same frequency band. Here in the high theta frequency band, like 8 hertz, it's commonly found between prefrontal and these sensory regions during working memory delay periods. So here's just an MEG study from recently that also sees this high theta, like 8 hertz phase synchronization between frontal and temporal regions during a working memory task when you're holding a real world complex objects in mind, in your mind's eye. And based on this work, we were really interested in trying to reproduce these effects, theta gamma phase amplitude coupling, in the relevant temporal areas, and theta phase synchronization between frontal and temporal areas in healthy young adults, and then ask the question, do the working memory deficits that have been observed for 3 decades as we get older in our 60s, 70s, and 80s, are they related to disruption, attenuation in these neurocoding schemes indexed by phase amplitude coupling and phase synchronization? So all that was just kind of to quickly bring you up to speed on what our hypotheses were for this particular study. So John Nguyen headed this up, a PhD student in the lab, and we started by just implementing this canonical delayed match to sample task, so with real world natural objects. So it comes in like this: you fixate a central point, there's this real world object presented, it goes away, there's a blank for 3 seconds, you see an object again, it's either the identical match or slightly different from the original, and you indicate with a button press whether it's the same or different. We also implemented this slick arousal, vigilance, attention control trial. We're borrowing from other researchers. It's identical to the memory trial, but here the task is at probe. You tell us whether it's tilted right or left from the vertical, and it's followed by a mask. So this allows us, the time of interest obviously is here during the delay period, and this just allows us to control for any stimulus or non-memory related neural activity during the delay by doing the memory versus control contrast. These are also blocked, and they're titrated by difficulty level. So we get the same number of trials and the same difficulty level between the control trial and the memory trial. We just look at behavior from the memory trials. This is, I think, 42 or 43 healthy older people and 42 or 43 20-somethings. And we just see what we'd expect based on the previous literature, that people are slower and less accurate at performing a task like this when you're older. So that's nice. Again, it's just a kind of reproduction of what we'd expect to see. While people were performing this standard task, working memory task, we had the EEG cap on them, and offline we measure phase amplitude coupling for a range of phase frequencies, amplitude frequencies, between for each subject, and use cluster-based permutation statistics to identify the memory-specific spatial spectral clusters for each group between the memory and the control trials. And when we do this, we find what others have found, this cluster of left temporal theta gamma phase amplitude coupling in younger people. Source analysis suggests that it's coming from a region in the left temporal cortex. We don't take inverse source modeling too seriously as telling us what the neuronal origins are of an EEG effect, obviously, but it gives us some confidence, some amount of confidence, and for example, potentially informs like future studies deploying like tools with greater spatial resolution, like monkey neurophysiology. We see an effect that has a peak 8 hertz phase frequency and 30 hertz amplitude frequency, pretty specific within this PAC space. This was kind of more surprising 4 years ago when this was first published. So, but no clusters were identified in older people. And again, this is just to show it's consistent with what we're finding in temporal cortex in humans in intracranial work, with a theta gamma PAC signature for working memory. We actually have subsequent studies showing a set size manipulation that I'll show you, which is commonly done in the working memory field to really nail down the working memory neural activity. When we seed the left temporal cortex theta band in the source points in the left temporal region and calculate this measure of phase-based connectivity, it's called phase locking value, for all source points in the kind of this proxy space and in cortical space, using MRI EEG beamforming, we find a cluster of theta PLV values in the frontal region in younger people, which was not present in older folks. When we do the same seeded connectivity analysis in the gamma band identified by the PAC analysis, we do find this kind of more short range gamma band connectivity in both age groups, between occipital and temporal regions. So that to us was interesting because it provided a dissociation that it was potentially this more longer range, low frequency connectivity that was attenuated or missing for older people, but not this more local processing, presumably short range gamma connectivity in the region where presumably the more storage per se is occurring. Again, this was kind of more the theoretical apparatus here is like more storage processing neural activity, and this is that more long range neural activity related to top-down control, monitoring, and coordination of the activity in the posterior region. And this was just consistent with this MEG study in healthy younger people who also found a similar result. Not an aging study, but it gels with their younger person finding using MEG, long range theta phase synchronization across the left frontal temporal cortex during a working memory task that specifically used complex real world objects as stimuli. So this is work that informed how we designed our non-invasive neuromodulation, our HDTACS. I've mentioned already that, so we use electric field models and really aim for increased vocality in these regions that we observed in our neural data. So we're taking this high definition approach. We also are frequency personalizing the stimulation. So for everyone they come into the study, there's like pre-experiment day, They do the delayed master sample task. We collect their EEG in an abbreviated version of that task. We compute the theta phase synchronization in source space. We grab the peak frequency, I think between like 2 and 12 hertz, so very broad theta band. And that's the frequency of stimulation that you'll get. And well, everyone's is a little different. There's quite an amount of variability across individuals, but a strong enough signal for us to detect it, detect the peak. So it's frequency personalized in that sense, in the sense of, it's keyed to a functional phase-based connectivity measure. And it's also a long distance synchronization. This is worth noting because there's a lot of, in this field, if you're not familiar, a lot of people target like a single region. So it's a little newer in the sense that we're targeting 2 regions simultaneously. And the beauty there is we hope, we think, we theorize that we can manipulate like the phasic relationships between these regions. And so here we're targeting the frontal and the left temporal region with HD-TACS. So we're doing this kind of multifocal, or goes by different names, synchrony modulation, in phase. The idea is that we're trying to facilitate the neural integration between these regions. And we're doing it in 0 phase. So it's 0 phase offset between these regions. So I can talk more about that, but we're not taking into account any axonal conductance delays. It's 0 phase stimulation. And we've had success in the past. We're kind of not shooting in the dark here. We've had some nice success in the past from this paper that showed this is kind of different cognitive process, different cortical targets. But we found nice evidence previously for this potentially bidirectional causal control over this, in this case, this circuit, this kind of medial lateral circuit in frontal cortex, by manipulating the phase angle of the alternating current that we're pushing and pulling through the brain. So it's really lovely here, I guess a paper I'm not really talking about, to see 0 degrees phase angle difference at this theta band coherence measure from the vertex to the DLPFC on the right, to upregulate that coherence or to downregulate it relative to a sham condition. Again, just based on whether it's in phase or anti-phase. So the in phase is 0 degrees phase offset. So we're pushing the current through and then we're sucking it out, in, out, with like 0 degrees phase difference between these regions. And the anti-phase is where you're kind of pushing it in and pulling it out like this. And it's thought to facilitate more destructively interfere with the dialogue between these regions. I'll actually touch on this more in a minute, but this first pass, we were just going for trying to integrate information across these regions using the in-phase stimulation approach. This is a double-blind, randomized, sham-controlled within subjects experiment, so everyone's their own control to enhance the statistical punchiness. Everyone receives 25 minutes of stimulation and then 50 minutes of post-stimulation period where they continue doing this task. It's paired with a sham condition where people receive the exact same stimulation, but it just ramps up and down at the beginning and end of the 25-minute period. This is the kind of gold standard in non-invasive neuromodulation research. So to simulate that itching, tingling, poking sensations that people often endorse on active days. And then we do a number of like visual analog scale questionnaires at the end to confirm people remain blind. Typically, this is a tool that we don't have much trouble keeping people blind with. So there's exceptions. Depends on where you're stimulating and how you're stimulating. But not in this case, because we're not really inducing visual phosphines, affecting retinal phosphines. And the stimulation order is counterbalanced across subjects. So actually, the way I just presented this was I just showed you actually all the sham baseline results first. And now I'm wowing you with the stimulation results. So incidentally, the young people, to control for demand characteristics, they also wore the whole apparatus and received the sham stimulation. And to our delight and surprise, we were able to sort of resurrect this cluster of phase-based connectivity in the frontal regions. again, this is not an activation here. This is actually a connectivity measure between left temporal and the frontal in the theta band. And we also were able to charge up the theta gamma phase amplitude coupling in the left temporal cortex. We did a number of directional coupling analyses too. And what we found is the theta activity in the front was more predictive of the theta activity in the back as opposed to the converse. And the theta activity in the back was more predictive of the gamma amplitudes in the same location versus the converse. So the idea we think is happening is the flow of information is going from the front to the back, and then it's pumping up those gamma amplitudes. So it's kind of like frequency remote way to stimulate and to move the information from the front to the back, and then to modulate gamma activity. But there's two separate measures. I should mention that when we look at spectral power, it's not changed. It's not changed in the frontal or temporal locations or in the frequencies of interest. I should mention that phase-phase coupling was also unchanged, that there's a degree of specificity. And I'll show you some further data to suggest that we really think we're latching on to a phase-based connectivity measure, and we're manipulating that stream of connectivity and not something else. The behavior came along for the ride, so we're able to improve people's memory performance on this simple laboratory working memory task. Obviously, we want to know how well it translates and generalizes, other stimuli, other stimulus modalities, lots of questions here, but at least for this pretty canonical delayed match to sample task, we're seeing like a 10% boost in people's working memory accuracy, but not their working memory speed. So, which is interesting. It didn't appear to be that we just had an effect on a simple speed accuracy trade-off mechanism, that we could make you better without slowing you down. So RT was not affected by stimulation, just the working memory accuracy. When we look at the theta gamma phase amplitude coupling against working memory accuracy relationships, we find that on an individual level, each data point's an individual. We find that people who had stronger coupling did better later in terms of their accuracy later down on that trial. So stronger coupling, stronger accuracy for young people. For old people, that relationship didn't hold up well at baseline, but then after stimulation, it looked a lot like the younger people's brain behavior correlation. So, you know, the suggestion here is that not only can we kind of resurrect, my battery's low, resurrect, where's our tech support? Resurrect these neural signatures important for memory, but that we can infuse them with functional significance. We're hoping the memory. I have one, but they weren't able to find a plug here. This is just to show that this is kind of just still preliminary because it's the N of 18. This is a subsequent study where just in healthy young people where we tried to get that bidirectional control. So apply the anti-phase stimulation like we did in 2017, and we're able to show that we can impair RT and accuracy on this task in healthy young people by exact same montage, but now just anti-phase, meaning it's 180 degrees phase offset between the two regions that we're pushing and pulling the current through. So that was cool. This was also something from that 2017 paper that we were able to show this bidirectional change in learning rate and adaptive control is a time estimation response learning task where we can measure how well you're learning a time interval over time based on feedback. We give you feedback or we take it away. And the non-feedback trials are here in white. You can see people getting better. There's interaction between feedback, trial, and block at baseline. And we could speed that up with the stimulation, or we can slow you down based on, again, this phase relationship that we were manipulating. We have the EEG neural data to help support that. We also looked recently at the meta-analytic level. Are we the only ones finding these cool results? Or what's it look like broadly for the whole field? So we did the biggest meta-analysis to date that we're aware of, over 100 studies, over 300 effects. And we are finding evidence for this in-phase improve, anti-phase impair relationship in the literature. So other researchers are obviously finding this as well. A lot of control analyses after this, basic principle findings. So one was just to ask how spatially and spectrally specific the stimulation effects are. And the upshot here is that we're able to reproduce an independent cohort of older people, the basic enhancement in working memory success on this task. But if we stimulate even within the same frequency band, but just not in a personalized manner, we don't see this boost. And if we stimulate even in the same spatial locations, but just individually, not at the same time, we don't see these benefits, which again suggests that it's really important to be modulating both these regions simultaneously in a phase-specific manner, as we see from the in-phase, anti-phase results. And this was, just a note of a little more rigorous than usual this test was. Typically in brain stimulation research, if you want to establish frequency specificity, you go to like a neighboring frequency. So for us, maybe that's delta or alpha. But this is actually within the frequency of interest. It's just not personalized. We used a uniform 8 Hertz stimulation and didn't see effect, which was pretty remarkable, I think. And then also spatially, typically go to like an atheoretical like location, like say the motor cortex, or this is within the regions of interest. It's just not simultaneous activation. So it's just, just want you to kind of appreciate that it was kind of more rigorous than it needed to be, but it turned out pretty interesting. Battery's probably going to die because I got many more slides. Yeah. No, it wouldn't reach, but if I could, I would have to move this onto the floor or something. She tried that and it wouldn't fit. I guess I have pretty clunky power. Apple gives you this big brick, so it didn't fit up here or down there. The wall would work if people are okay with that. I don't know if, don't have an adapter.
Sometimes they'll have like a.
Yeah. I don't think Dell, I don't know if.
This is too far away.
Yeah. I can put this on the floor, if that's going to work.
Doesn't fit in here. This is too big.
No, you can try. Maybe yours is smaller than mine. Oh yeah, I have a slightly larger one. Ah, there you go. Beautiful. Thank you. Sorry about that.
Should be able to reach. Why is that not like? Is it charging? That one. It is. Pick it.
Is that? No, I don't. Is that the charge sign? Okay. Great. Thank you. So, yeah, I mean, the basic science insights here, I think we make some inroads on the parameters of this presumably mechanism for working memory accuracy, which and parameters of frequency, space, and phase. Also, from the clinical perspective, it allows us to get some grip on what kind of parameters we need to think about when we design stimulation to be effective. And the work obviously connects with theories of like cortical disconnection in the aging literature that suggest these functional and structural connectivity deficits underlie cognitive decline, and theories on prefrontal cortex and aging. But I want to move along. There's time to tell you about some newer directions of the research of how we're taking this work. So one direction is to get into a little more granularity with these kind of subroutines of working memory. So, referred before, I showed you like this cartoon of like encoding, maintenance, and retrieval processes, but we know, it might be like deletion, copy, there's different like kind of myriad processes within the working memory system. And one that we're interested in that's really not well researched is deletion clear out process, like the end of a trial. Oh, I'm sorry. That is what we're doing. And I have those data. This slide's different. Sorry. So here, postdoc Wen is interested in more executive control processes during the working memory retention interval. We've known from the aging literature that this is where older people have problems. So she had a nice manipulation of distraction and interruption. So here, people are just storing the orientation of these bars in mind. And then there's a control trial where you just get these dots that are static. And then other trial types where the dots are moving, it's kind of distraction during the delay period. And another condition, it's not another condition where the dots are also moving, and then they make this sharp movement in one direction. You need to respond with a button press as to whether you identified that. So it's more of like a dual task or interruption, we're calling it. And then you get probed as to what target that we want you to give this continuous behavioral report of, what's its orientation. And so this is still preliminary with N of 25, but we can see this age-related interruption effect, which is kind of nice with the deviation between people's report and the target orientation, reaction time, and accuracy on this task. Using, training a classifier with people's EEG data on the different orientations, different trials. We can see in young people, the control and the interruption conditions, we can, the decoding is above chance accuracy. And for older people, that's only true of the control trial, not the interruption trial. We see evidence of a theta response in frontal regions in younger people, which is absent in older people. And then also the presence of that theta phase synchronization signature, again, from frontal to left temporal regions. That was present only in young folks. It was pretty frequency specific. Something happening here in the beta, gamma, but the largest effect here is in the theta band between control and between younger and older people. The frequency of this theta phase synchronization measure. And then Wen also put together a very similar frontal left temporal HDT-ASCS in-phase procedure and was able to show so far behavioral benefits in terms of precision and deviation for this task. So that was a nice reproduction, or at least conceptual replication of our 2019 findings in this more like cognitively interesting task. That's one thing that we're following up on. We're doing this in the scanner now as well. We just got funding to do an MCI and Alzheimer's study with our neurologist partners. And this is taking the same delayed match to sample task, but just adding a set size manipulation and applying the stimulation in healthy older people and people with mild Alzheimer's disease. We see the nice set size effect using the K capacity measurement in healthy older people, but this deficiency in people with Alzheimer's across set size, especially for larger set sizes. And this is just N of 10, so it's just a teaser data that went into the grant, showing when we use that same brain stimulation approach, we can boost people's working memory success. Really, it's where they need it the most, in these higher set sizes where there's potentially more room for growth. And this was advanced MCI in early Alzheimer's patients, just a sample size of 10, but pretty encouraging. So we're doing a proper clinical trial now with a large sample with Andrew Budson as the neurologist collaborator. Should be paying attention to time, but I want to talk about another paper real quick here, also about working memory and long-term memory. So we were, serial position curve effects, so these, really lovely indices, behavioral indices of working memory, long-term memory, at least from this dual store framework. There's other frameworks. I'm going to just stay in a classic dual store framework. And right, so this is if I read you a list of words, there's a behavioral advantage for remembering the words. You're freely recalling as many as you can after I read this list of words to you. You have an advantage for the words early on the list and later on the list, deemed the primacy, recency effect. There's a fair amount of research, almost 100 years of experimental psychology and neuropsychology research, which is pretty compelling. People with selective deficits just of working memory, so you see a selective abolition of recency but not primacy, and the opposite too, selective deficits in primacy with long-term memory dysfunction, but with working memory intact and recency intact. Really nice kind of double dissociations in neuropsychology. The functional neuroanatomy is fairly well characterized. These effects, again, have just been around for so long. We know the inferior parietal lobule is a major player in recency. DLPFC is a major contributor in primacy. What we don't know, what we'd like to know in our lab is more about these kind of neurocomputational mechanisms. Are there frequency rhythmic mechanisms in these segregated regions? And if there are, then potentially we could selectively improve memory just in this domain or just in that domain. So obvious clinical implications, but just also basic science insights as to what is the relevance for timing in the brain. So Shrey, PhD student, now postdoc in the lab, headed this work up. This was a random double-blind, sham-controlled series of experiments with over 150 participants aged something like over 65, like 65 to 88, something like that. And the goal was to see if we can modulate low frequency activity in parietal cortex and change recency, working memory, auditory verbal working memory content, whether we could entrain high frequency activity in DLPFC and see changes in long-term memory, auditory verbal content, the primacy effect. And so Shrey and I designed these two novel HDT-ACS protocols, and we were especially interested in sustained effects here. So we've done this a few times. It's still kind of new in the last few years of applying the stimulation over repeated days. So that's what we did here. It was 4 days of repeated stimulation consecutively to see, and measurement of memory performance every day within the intervention, which is also quite novel, and 1 month out. So this is a pretty busy slide, but the first experiment is what I described. The second experiment, we want to really test the specificity of our hypotheses. So we just switched the entrainment frequencies. So high frequency parietal, low frequency prefrontal. And we found that, and here's to orient you a little bit, probability of recall on the y-axis, the primacy, 3 middles, and recency on the x-axis. Each bar is kind of moving chronologically through the intervention. So the white bar is pre-intervention baseline. And then you see day 1, day 2, day 3, as you get warmer and warmer. And the last darkest bar is the 1 month post test using this free recall. Incidentally, these were words that we took from the Penn Kahana lab reservoir of data, so they're all normalized on all these various features and cleaned up. Thank you, Mike Kahana. And so he was a really pioneer in this field as well. And so the upshot here is you can see it outlined in the red boxes as we apply the low frequency TACS to the parietal cortex. You can see actually doesn't have any effect. So if you just did a one-shot stimulation, you'd be like, oh, what, TACS doesn't work. Actually needed to ramp up over the days and accumulate, and didn't come online to be statistically significant relative to sham until, what was it, day 3, and then day 4 is even higher. And then you can see it kind of level off and start to sink back down toward baseline levels, though never approaching quite baseline levels quite yet at the 1-month time point. So we have a really long-lasting effect, but we had to stimulate every day for 4 days in a row, and the effect didn't come online until day 3 in terms of the IPL theta modulation. In terms of DLPFC gamma, it came on a little sooner, but still it wasn't immediately observable on day 1, but it was statistically significant day 3, day 2, day 3, day 4, and then 1 month post. Presumably, the benefits come down to baseline levels if we kept tracking memory performance out longer than 1 month. But 1 month is a long time for this, extremely weak electrical current, although applying it repeatedly. And so the design was, here are the electric field models. So this is application of theta and intraparietal lobule. So really big place. We were really just going more for intensity than focality as this kind of proof of principle study. So, but still better spatially resolved than the conventional TACS approach. And so this was just trying to entrain neural activity in parietal cortex in the theta band versus entraining neural activity in the gamma band in the left DLPFC. Yeah, right. Yeah. And we also, in experiment 2, just saw a degree of specificity. So if we switch the entrainment frequencies, we don't see the same effects. Theta in the DLPFC and gamma in the IPL didn't show results here. So, we have a double dissociation between frequency and space on the one hand, and primacy and recency on the other hand. Incidentally, in terms of basic science, we've known that for a long time in terms of space. Like, there's a lovely TMS study doing with the same targets, showing a double dissociation between primacy and recency and DLPFC and IPL. What the contribution here is really the frequency information. And then here are some additional points. So if you look at the rate, it's not commonly done where you assay behavior like during a stimulation intervention. It's more commonly like pre versus post. So here we actually have this opportunity to look at how memory was changing on each day of the intervention. If you look at just the rate of benefits, rate of improvement, that slope, it was predictive of the memory benefit one month later, which was kind of cool. Maybe in some future application down the line in the clinic, you can imagine having understanding about responders, non-responders based on how they're performing during the intervention, how long and how potent the effect will be later on. Here's also something maybe interesting to stimulation researchers as we're trying to identify the relevant parameters of effective stimulation. This was a look at the baseline cognitive performance of each individual and to see how predictive it was. So people who have poor baseline cognitive functioning benefited the most. We typically see that relationship, but it was nice. It was assessed here with the MoCA, and it predicted how much memory benefit they would experience during the intervention and how large the effect would be 1 month later. Just taking snippets from this meta-analysis we recently did because there was a subset in the study just concerning older people. So if we just look at all the TACS cognitive studies in older people to date, there's several dozen. And this cuts across, attention, working memory, executive function. Didn't have enough, power, enough studies to reliably infer, estimate effect sizes for each domain specifically. So this is all pooled together, but it looks encouraging. We're seeing effects to like the modest to moderate range in enhancing these various cognitive components in older people at the meta-analytic level. So I think I'm out of time. I'd like to leave some time for questions. Interaction's always more interesting. So thank you.
Really interested in sort of longer term effects. So there's like a greater study testable. And I wonder if you've looked at whether stimulation is most beneficial when it's delivered at encoding versus retrieval or both.
Yeah, that's a great point. That's actually what Shrey is doing right now in the lab. We have a task that separates encoding and retrieval, and we're stimulating at those individual, like separately for those periods of the task. And there's some nice neuroscience about, we have a number of ideas. Like one is like low versus high gamma in encoding versus retrieval. There's some evidence for that distinction. And then also distinctions between theta and gamma coupled, like their exact phase structure, whether the gammas land on the peak versus the trough. There's some nice work suggesting that that's actually an important feature of the mechanism that also distinguishes encoding from retrieval. And that's something we're doing in the lab now. We can actually make these more complex waveform TACS protocols, introduce theta gamma coupling in the brain. We actually have some really nice evidence for that, but it's in like more low vision. We just submitted for a grant and looking at 3D shape perception via motion parallax, how you recover depth from motion and contrast sensitivity. That's another talk, but it involves these interacting high and low frequency waves that have been kind of shown in neuroscience. And now we have this tool where we can construct it and introduce it in target regions with TACS. But yeah, that's a great question. That's what we're interested in right now. Hi.
This is something, a question that probably people who do this is obvious, but I got a little confused. When you talked about the working memory study, you presented these two mechanisms, like motifs, right? One was theta gamma, I guess the amplitude modulation, essentially, and then one was more this phase coupling. And then something I didn't quite understand, it seemed like your manipulation for two brain areas, the frontal and I guess parietal region, was kind of phase coupling. But then the result was a theta gamma, with the effects were theta gamma modulation. So maybe
yeah, you're trying to like integrate the two studies. Oh yeah, because the first study was about theta gamma phase amplitude coupling in temporal cortex and that long range theta phase synchronization between prefrontal and temporal. Yeah. In the first study. Yeah. Yeah, I mean, we think that's the mechanism for how the prefrontal region is communicating with that posterior region.
But didn't you do that and then you got more of the theta gamma?
Yeah, we got more of the theta gamma. So it's kind of this this dual mechanism, or maybe it's one mechanism, but it's the frontal and temporal regions synchronizing. And then subsequently, we think from the directed coupling evidence, we think subsequently that then the theta back here is driving the gamma amplitudes in the same region. So it's kind of this like multi-part process. But we're synchronizing kind of near, like in the causal chain of events, we think more early on the front end, and then it's subsequently having these effects in that local theta gamma coupling relationship.
The other thing I wanted to ask you is synchronizing the phase difference, even though there should be some presumably delay because of axonal communication. So why?
Yeah, no, it's interesting. I mean, it's not even, Yeah, there's a whole slew of research from animal models showing like 0 degrees phase offset and behaviorally meaningful outcomes with 0 degrees phase offset. It's kind of an interesting question on its own. But yeah, apparently you don't have to, from our perspective, like human network and stimulation, we're not having to do this meticulous accounting of phase lag. Although we're interested in this. Actually, if you look at the modeling of these simple like single cell spiking models, just came out like a year ago from Engel's lab. Bettina Schwab is the first author in our image paper, and it shows that when you do look across the phase lags, you see the plasticity is like optimal at like 0 degrees phase lag, and then it just rapidly drops off like with like an exponential. And then once you get to like these very like 5 millisecond, 10 very small milliseconds, you're almost at like 0 again. So it's in their model, 0 was still optimal, but there's a very fast fall off. So we recently, it's not published, put like 2 stimulators on someone's head. These are schizophrenia patients and healthy controls, just like small samples. And we did like 0 phase and then slight offsets like 5 millisecond, 10 millisecond. And we could see like parametrically like the effect just like wane as you move away from 0. So that was cool. If we get funding, the grants in, then we could follow up and publish that. But based on the modeling and then based on the animal work, apparently 0 phase lag is a thing, and it's optimally where we want to be. It's kind of like intuitively, it kind of violates our common sense intuition. Like, why wouldn't you need to account for these? I mean, they're very small axonal conductance delays, but if you account for it, why shouldn't it be better? But don't have a good, I guess the good response to that would probably be in the biophysics of cells.
In active older adults, we see that segregation is an effect of child older adults and similarly younger. So how long is that?
The increased theta gamma coupling in older people. By the way, we were also able to increase theta gamma coupling in younger people who were like the weak couplers. That was an experiment I didn't mention. We invited back all the people from experiment 1, just who were like the weakest couplers, and we were able to boost their coupling and working memory performance. How long does it last? So we binned the data maybe 8 minutes, something like that. It was 2019. Like these smaller bins during the 50-minute recording period after stimulation was over, we binned it so we could look at like the neural behavioral dynamics over the 50-minute period. And so they remained, it was like more stable than I expected. And it just remained throughout the 50-minute period. So short answer is we don't know. That particular procedure, we haven't gone and checked out, days and weeks and months. We're doing a lot of that now. Like we went back to that 2017 paper, which I alluded to, where we changed like executive function, learning, how well you adapt your behavior after you make an error on that time estimation learning task, and the bidirectional regulation of theta coherence up and down, whether it's in phase or anti-phase. We now personalized that, and we looked out 2 weeks. And with the personalized version, that was also repeated for 5 days in a row. So it's not a one-off, one-shot TACS, but that lasted for 1 week. So there's a lot of unknowns about the duration of the effects. It's actually one of the aims of a grant that we have. So we should be a little more knowledgeable of the outcomes, the duration of the effects soon. Yeah, it's a good question. Definitely longer than 50 minutes and probably less than 1 week. Yeah. That's a good like kind of clinical applications kind of question. Yeah. I guess it's within our reach to do something like that. A lot of the thought in the lab is just like, how is this working? What are the like physiological mechanisms? But then you're right, when we see these effects that last, you get this whole world of questions that comes along, and they have a lot of clinical flavor, and that tends to be like of less interest to people in my lab. But we get all that because we get invited to like psychiatry and neurology conferences, and yeah, don't know, don't have a, it's hard to motivate people in my lab if they're not interested in doing something. They, you know.